Bioaccumulation of lead and nickel using Fenugreek in highly contaminated soil. Heavy metal contamination is an increasing worldwide environmental concern, and phytoremediation is one of the best solutions as it remediates the heavy metals from the contaminated sites. One of the mechanisms of phytoremediation is phyto extraction, in which plants are used to extract the heavy metals from contaminated soil. There are two types, natural and induced phyto extraction. In induced phyto extraction, ethylene diamine tetraacetic acid has been widely used as it has high extraction efficiency. Salicylic acid, it is an endogenous growth regulator and it have resistance to toxic action of heavy metals. The present study aims to analyze and evaluate the effect of EDTA and salicylic acid on the accumulation of lead and nickel by fenugreek plants in contaminated soil and determine the distribution of lead and nickel in the stem, leaves and roots of fenugreek plants. The study was done under the field condition at the micromodel experimental site of IIT Delhi. These are the physiochemical properties of the soil. First, 20 seeds of fenugreek were sown in pots which have soil, farmyard manure and sand in ratio of 2:1. In chemical treatment, lead, nickel, EDTA and salicylic acid were added as per the design treatment. Plants were watered daily using tap water. Seed germination started after the 7th day of sowing and plants were thinned to 3 plants per pot after 30 days of sowing. Samples of plants were taken 7 days and monthly after chilling treatment. At the end of the experiment, the plants were harvested, washed accurately, and the aerial part was divided from the root and the two parts were analyzed separately to determine the metal content. Some observation was also made to determine the localization and translocation of metals in plant tissues. This is the design treatment where T0 is the control, T1 is 100 mg per liter lead, T2 is 800 mg per liter lead, T3 800 milligram per liter nickel, T4 is amount of lead and nickel 800 milligram per liter. T5 is the addition of 100 millimolar EDTA along with lead and nickel metal, while T6 is the addition of 1 millimolar salicylic acid along with lead and nickel metals. Soil and plant sample were analyzed with the help of standard methods. SAM and EDX spectra was generated with the help of standard methods. Statistical analysis was done in Minitab version. Results. Plant growth. Application of salicylic acid with lead and nickel treatment helped in reducing the inhibitory effect of these metals on seed germination along with increased dry weight. There was no significant negative effect on plant growth observed in the presence of salicylic acid. Plant metal accumulation. Lead concentration increased in both organs of the plant, but it decreased with T5 and T6 treatment. Translocation factor was also evaluated, which is the ratio of shoot metal concentration over root metal concentration, and it was highest for T2 and lowest for T5. Salicylic acid helped in the translocation of lead and nickel in the aerial parts of the plants. Some observations are lead accumulation was highest in root, while nickel accumulation was also highest in root at T5, and in stem it was highest at T4 and in leaf at T6. Accumulation of lead and nickel affected the presence of calcium and iron, as calcium was highest in T4 treatment and iron was highest in T1 treatment. The presence of EDTA caused decrease in nickel accumulation in stem. These are the element accumulation in plant tissues of Fenugreek treated with different lead and nickel treatments. And these are the SAM and EDX spectra of lead treated leaf samples, stem sample, and root samples of different EDTA, nickel, and salicylic acid treated plants. SAM micrographs displays various structural effects such as reduced number of stomata on the surface of the leaf at 800 mg per liter of lead treatment. Nickel was found to accumulate preferentially in the upper leaf epidermal cells. SAM micrographs of roots revealed accumulation of lead and nickel mainly in cell walls, suggesting transport via the apoplastic pathway. The epidermis provided a major barrier to the transport of lead only. Now come to the conclusion. The results indicate that the remediation of lead and nickel by Fenugreek can be enhanced by the addition of EDTA. In the presence of EDTA, plant growth was significantly inhibited, whereas in the presence of salicylic acid, plant growth was as good as the control. SAM confirmed the presence of lead and nickel inside tissues of the plant. SAM analysis revealed that in the presence of EDTA, the deposition of lead particles was predominantly in vascular tissues of the stem and leaf. In roots, deposition of lead was in the steel region. However, in the presence of salicylic acid, lead particles were more concentrated in the cortical region. However, further experiments are still needed to determine the actual mechanism involved in lead and nickel uptake by Fenugreek. These are the references. Thank you.